Entamoeba histolytica, Wikipedia audio. Entamoeba histolytica is an anaerobic parasitic amoebozoan, part of the genus Entamoeba. Predominantly infecting humans and other primates causing amoebiasis, E. histolytica is estimated to infect about 50 million people worldwide. Previously, it was thought that 10% of the world population was infected, but these figures predate the recognition that at least 90% of these infections were due to a second species, E. dispar. Mammals such as dogs and cats can become infected transiently, but are not thought to contribute significantly to transmission. The word histolysis literally means disintegration and dissolution of organic tissues. The active stage exists only in the host and in fresh loose feces. Cysts survive outside the host in water, in soils, and on foods, especially under moist conditions on the latter. The infection can occur when a person puts anything into their mouth that has touched the feces of a person who is infected with E. histolytica, swallows something, such as water or food, that is contaminated with E. histolytica, or swallows E. histolytica cysts picked up from contaminated surfaces or fingers. The cysts are readily killed by heat and by freezing temperatures, and survive for only a few months outside of the host. When cysts are swallowed, they cause infections by existing in the digestive tract. The pathogenic nature of E. histolytica was first reported by Losch in 1875, but it was not given its Latin name until Fritz Schaudin described it in 1903. E. histolytica, as its name suggests, is pathogenic. Infection can be asymptomatic or can lead to amoebic dysentery or amoebic liver abscess. Symptoms can include fulminating dysentery, bloody diarrhea, weight loss, fatigue, abdominal pain, and amoeboма. The amoeba can actually bore into the intestinal wall, causing lesions and intestinal symptoms, and it may reach the bloodstream. From there, it can reach different vital organs of the human body, usually the liver, but sometimes the lungs, brain, spleen, etc. A common outcome of this invasion of tissues is a liver abscess, which can be fatal if untreated. Ingested red blood cells are sometimes seen in the amoeba cell cytoplasm. Transmission. Poor sanitary conditions are known to increase the risk of contracting amoebiasis E. histolytica. In the United States, there is a much higher rate of amoebiasis-related mortality in California and Texas, which might be caused by the proximity of those states to E. histolytica endemic areas, such as Mexico, other parts of Latin America, and Asia. E. histolytica is also recognized as an emerging sexually transmissible pathogen, especially in male homosexual relations, causing outbreaks in non-endemic regions. As such, high-risk sex behavior is also a potential source of infection. Although it is unclear whether there is a causal link, studies indicate a higher chance of being infected with E. histolytica if one is also infected with HIV. The E. histolytica genome was sequenced, assembled, and automatically annotated in 2005. The genome was reassembled and renotated in 2010. The 20 million base pair genome assembly contains 8,160 predicted genes. Known and novel transposable elements have been mapped and characterized, functional assignments have been revised and updated, and additional information has been incorporated, including metabolic pathways, gene ontology assignments, curation of transporters, and generation of gene families. The major group of transposable elements in E. histolytica are non-LTR retrotransposons. These have been divided in 3 families called elines and essenes. Eline1 encode an endonuclease protein, which have similarity with bacterial restriction endonuclease. This similarity with bacterial protein indicates that transposable elements have been acquired from prokaryotes by horizontal gene transfer in this protozoan parasite. In the vast majority of cases, infection is asymptomatic and the carrier is unaware they are infected. However, in an estimated 10% of cases E. histolytica causes disease. Once the trophozoites are excisted, they colonize the large bowel, remaining on the surface of the mucus layer and feeding on bacteria and food particles. Occasionally, and in response to unknown stimuli, trophozoites move through the mucus layer where they come in contact with the epithelial cell layer and start the pathological process. E. histolytica has a lectin that binds to galactose and N-acetylgalactosamine sugars on the surface of the epithelial cells. The lectin normally is used to bind bacteria for ingestion. The parasite has several enzymes such as pore-forming proteins, lipases, and cysteine proteases, which are normally used to digest bacteria in food vacuoles but which can cause lysis of the epithelial cells by inducing cellular necrosis and apoptosis when the trophozoite comes in contact with them and binds via the lectin. Enzymes released allow penetration into intestinal wall and blood vessels, sometimes onto liver and other organs. The trophozoites will then ingest these dead cells. This damage to the epithelial cell layer attracts human immune cells and these in turn can be lysed by the trophozoite, which releases the immune cell's own lytic enzymes into the surrounding tissue, creating a type of chain reaction and leading to tissue destruction. This destruction manifests itself in the form of an ulcer in the tissue, typically described as flask-shaped because of its appearance in transverse section. This tissue destruction can also involve blood vessels leading to bloody diarrhea, amoebic dysentery. Occasionally, trophozoites enter the bloodstream where they are transported typically to the liver via the portal system. In the liver a similar pathological sequence ensues, leading to amoebic liver abscesses. The trophozoites can also end up in other organs, sometimes via the bloodstream, sometimes via liver abscess rupture or fistulas. In all locations, similar pathology can occur. E. histolytica may modulate the virulence of certain human viruses and is itself a host for its own viruses. For example, AIDS accentuates the damage and pathogenicity of E. histolytica. On the other hand, cells infected with HIV are often consumed by E. histolytica. Infective HIV remains viable within the amoeba, although there has been no proof of human reinfection from amoeba carrying this virus. A burst of research on viruses of E. histolytica stems from a series of papers published by Diamond et al. From 1972 to 1979. In 1972, they hypothesized 2 separate polyhedral and filamentous viral strains within E. histolytica that caused cell lysis. Perhaps the most novel observation was that 2 kinds of viral strains existed, and that within one type of amoeba the polyhedral strain had no detrimental effect but led to cell lysis in another. Although Mattern et al. Attempted to explore the possibility that these protozoal viruses could function like bacteriophages, they found no significant changes in Entamoeba histolytica virulence when infected by viruses. However, no newer published research has been conducted on this species since. Diagnosis is confirmed by microscopic examination for trophozoites or cysts in fresh or suitably preserved fecal specimens, smears of aspirates or scrapings obtained by proctoscopy, and aspirates of abscesses or other tissue specimen. A blood test is also available but is only recommended when your healthcare provider thinks that your infection may have spread beyond the intestine to some other organ of your body, such as the liver. However, this blood test may not be helpful in diagnosing your current illness because the test can be positive if you had amoebiasis in the past, even if you are not infected now. Amoebic intestinal ulcer caused by E. histolytica. Trophozoites of E. histolytica with ingested erythrocytes. Risk factors. E. histolytica cyst. Immature E. histolytica cyst. Multiplication by binary fission. E. histolytica drawing. There are a number of effective medications. Generally, several antibiotics are available to treat Entamoeba histolytica. The infected individual will be treated with only one antibiotic if the E. histolytica infection has not made the person sick, and most likely be prescribed with 2 antibiotics if the person has been feeling sick. Otherwise, below are other options for treatments. Intestinal infection. Usually nitroimidazole derivatives are used because they are highly effective against the trophozoite form of the amoeba. Since they have little effect on amoeba cysts, usually this treatment is followed by an agent that acts on the organism in the lumen. Liver abscess. In addition to targeting organisms in solid tissue, primarily with drugs like metronidazole and chloroquine, treatment of liver abscess must include agents that act in the lumen of the intestine to avoid reinvasion. Surgical drainage is usually not necessary except when rupture is imminent. Genome. Pathology. People without symptoms. For people without symptoms, non-endemic areas should be treated by paromomycin, and other treatments include diloxanide furoate and iodoquinol. There have been problems with the use of iodoquinol and iodochlorhydroxyquin, so their use is not recommended. Diloxanide furoate can also be used by mildly symptomatic persons who are just passing cysts. Pathogen interaction. Diagnosis. Treatment. Meiosis. In sexually reproducing eukaryotes, homologous recombination ordinarily occurs during meiosis. The meiosis-specific recombinase, DMC1, is required for efficient meiotic HR, and DMC1 is expressed in E. histolytica. The purified DMC1 from E. histolytica forms presynaptic filaments and catalyzes ADP-dependent homologous DNA pairing and DNA strand exchange over at least several thousand base pairs. The DNA pairing and strand exchange reactions are enhanced by the eukaryotic meiosis-specific recombination accessory factor HOP2-MND1. These processes are central to meiotic recombination, suggesting that E. histolytica undergoes meiosis. Several other genes involved in both mitotic and meiotic HR are also present in E. histolytica. HR is enhanced under stressful growth conditions concomitant with the upregulation of HR-related genes. Also, UV irradiation induces DNA damage in E. histolytica trophozoites and activates the recombinational DNA repair pathway. In particular, expression of the RAD51 recombinase is increased about 15-fold by UV treatment.